So our next speaker this morning is Savante Paabo. Savante has developed techniques and approaches that allow DNA sequences from archaeological and paleontological remains to be sequenced. This has allowed ancient DNA from extinct organisms, humans, animals, and pathogens to be studied. His research group has determined high-quality Neanderthal genomes, allowing for the reconstruction of the recent evolutionary history of our species and the realization that Neanderthals contributed DNA to present humans who live outside of Africa. By studying DNA sequences from a small Siberian bone, they discovered the Denisovans, a previously unknown hominin group, distantly related to the Neanderthals. Savante also works on the comparative and functional genomics of humans and apes, particularly the evolution of genetic features that may underlie aspects of traits specific to humans. Savante has received several honorary doctorates and scientific prizes and is a member of numerous academies. He is currently director at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. And Savante will be talking this morning on Neandertal genomics. So, over to you.
Okay, thank you very much for the invitation to come to this amazing place. That, as you will hear in the next talk, we actually even work a little bit on remains from here. So I wanted to expand maybe the topic a little bit to not just Neandertal genomics, but archaic genomics, including Denisovans. And as you know then, our big interests are Neanderthals and Denisovans, and sort of the forms of hominins that modern humans meet when they come to Eurasia. And as you also know, there have been lots of opinions around in the paleontological community about the relationship between present-day people. This was the thing, it doesn't point to the screen. That's embarrassing. Let's see. This thing works? Yes. Okay. You'll have to tell me if you don't hear me. So about what relationship between present-day people and Neanderthals and groups in Asia, if there was an absolute replacement, as many people were thinking, particularly in the molecular field a couple of years ago, or if there were different forms of relationships between now extinct forms of humans and present-day humans. And as you also heard alluded to, we sort of got a first chance to look at this in the early 90s when we studied the type specimen of the Neanderthal, at that time focusing on the mitochondrial genome that you know is this tiny part of the genome that is maternally inherited, so from mother to offspring, focusing on a particularly variable part of the mitochondrial genome, at that time using this cumbersome technique, polymerase chain reaction, where you multiply short fragments of the sequence you're interested in, clone it and sequence many, many DNA molecules, and believe the substitutions that are then consistently there. And what came out of that was that the mitochondrial genome of Neanderthals go back to common ancestor in the order of 500,000 years ago, shared with the mitochondrial genomes of present-day people. And all present-day mitochondrial genomes go back to common ancestor 100, 200,000 years ago. So in terms of sort of a scheme where you would say on one extreme you have total replacement, where modern humans receive nothing from earlier forms of hominins, to total continuity. In terms of the mitochondrial genome, it's total replacement. There is no one running around today with a mitochondrial genome from a Neanderthal. But as you also know, the big story is in the nuclear genome that contains vastly much more information, and that's inherited both from mothers and fathers to offsprings. And the chance to study that then came in the beginning of the millennium with high throughput DNA sequencing technologies, where you can sequence millions and billions of molecules rather inexpensively. So what you can do is extract DNA from a fossil, sequence all the DNA that's in there, make your own little database and compare the DNA fragments you have to the human genome, bacterial genomes, and other genomes. So we were then very lucky to be funded for 5 years to work on improving the techniques to extract the DNA from the bone and for converting this DNA into form that you can sequence and study. And we got a lot better on that. And one also learned a lot of things in that process. So for example, when you now sequence entire molecules here, you are able to see the very ends of your DNA molecules. And it turned out that if you walk in from the very tip of the molecule inwards, you will find that one of the 4 bases that make up the DNA, the Cs, very frequently appear at Ts at the first position. So up to 30–40% of all the Cs look like Ts. And that turns out to be due to particular chemical damage that also affects amino acids and other molecules. This is a loss of this amino group, so deamination. So this C turns into another base that naturally doesn't occur in the DNA, but that is read by the enzymes you use when you sequence as a T. So there are apparent C to T substitutions due to that these uracils are there. And that will become very important in the next talk that you will hear, actually. But the big advantage of this, I should say, before just alluding to that, is that this is then an effect that takes thousands of years to accumulate in the DNA, so it can be used as an indication that the molecules that carry this C to T substitutions are actually ancient and not modern contaminants. So back in 2010 then, we had the first draft, rough version of the Neanderthal genome. Came from 3 bones from Croatia, from Vindija Cave, from 3 different individuals. So sort of a composite of 3 Neanderthal individuals, where one had then sequenced about 1 billion DNA molecules and then mapped them to the human genome that was then available, taking into account that there are these particular C to T changes towards the ends of the molecules. So that was then a rough draft. So we had covered only a little over half of the genome, once or more. But one could begin to ask questions anyway. And of course, one question was this: what happened when modern humans met Neanderthals? Did one mix or not? And if modern humans would then have mixed with Neanderthals so that Neanderthals contributed to present-day people in Europe, an expectation of that would, of course, be that Europeans today would share more genetic variants with Neanderthals than people in Africa, where there have never been Neanderthals. So to address that, could then do was to use the Neanderthal genome and compare to present-day people. And to just illustrate this idea, if we take 2 African individuals, we can find the places in the genome where they differ from each other. And then we take the Neanderthal genome and we see how often does a Neanderthal map one African or the other African. That should really be 50-50, right? Because since Neanderthals have never been in Africa, there's no reason to assume that one African is closer to the Neanderthal than another African is. And indeed, statistically speaking, it's sort of 50-50 of matches to one African to the other. To my surprise, actually, at the time, when we then did this instead with a European individual and an African individual, we found significantly more matching to the European individual than the African individual, suggesting that there could have been a contribution there. So even more surprising, when we took a Chinese individual, where most people would perhaps say there have never been Neanderthals in China, we found more matching. Papua New Guinea, where I think everyone agrees there have never been Neanderthals, again. So no matter what individual outside sub-Saharan Africa we look at, we find this additional contribution from Neanderthals. That led then to this idea that if early modern humans mixed with Neanderthals and then became the ancestors of everybody outside Africa, they could carry with them this Neanderthal component out to the rest of the world. To the extent that no matter where you look outside sub-Saharan Africa, 1 or 2% of the genomes come from Neanderthals. It's a much more complex picture like this, as you will see in a minute, but sort of on a coarse bird's eye view, that still holds up. What has then happened is that it has become possible to generate high quality genomes from Neanderthals. That particularly comes from the collaboration with Russian colleagues, particularly Anatoly Derevyanko and his team that excavates, as many of you know, at many places in Siberia. And particularly then this site, the Denisova cave in the Altai Mountains, this place. Where they in 2010 found this toe bone that turned out to come from a Neanderthal. And by that time, we had also improved our techniques a lot in retrieving many DNA sequences from tiny amounts of badly damaged DNA. One of the key improvements was this methods where you separate the two DNA strands. As you will know, the DNA is a double-stranded molecule. The same information is encoded on both strands in a complementary form. And most methods rely on using this double-stranded DNA, converting into its form that you can sequence by adding the synthetic pieces of DNA in the end. What Matthias Meyer came up with in our department was to start by separating these two strands and then converting each of the strands to a molecule that you could sequence. So each double-stranded molecule have two chances to end up in the libraries we sequence, one for each strand. And that turned out to be very crucial because often you have chemical modifications on one strand that makes a molecule to get lost and you can't study it. But then the other strand would still have a chance. So with this and other improvements, it was then possible to go from a version of the Neanderthal genome where you had a few fragments here and here, there on a chromosome like this, to one where every position is covered on average 30 or up to 50 times. So that was a good Neanderthal genome then, that you can then compare in detail to present-day genomes, for example, in Europeans. So if you do this, in this particular case, we compared Neanderthal genome to 173 Europeans. And you can then identify quite accurately longer pieces of DNA that are very similar to the Neanderthal genome and sort of come from Neanderthals. So on average, Europeans will then carry 23 million base pairs that come from Neanderthals. And they are distributed in fragments of around 50,000 base pairs scattered around the genome. So for example, there will be on average in any European person 58 amino acid changes that come from Neanderthals over to present-day people. So you can then on a certain chromosome say, mark in red here fragments that come from Neanderthals, and you will see that they add up on average to 1 to 2%, but many people carry different fragments from Neanderthals. So you can actually go from person to person in this room, for example, and try to puzzle together the bigger sections of the Neanderthal genome from present-day people. And that's indicated in blue here, and that adds up to something like 40–50% of the Neanderthal genome is still around walking on two legs today, so to say. So what has then happened is that we've been able to generate more high-quality Neanderthal genome. The next one is then from this site in Croatia, the India cave. From this bone here, we've actually had to take several samples, as you see. And you can then ask how people who live in Europe today, are they more closely related to this Neanderthal from Siberia or to the Neanderthal from southern Europe? By just saying, again, sharing of sites, just as I indicated with the Europeans and Africans earlier. And you will find, not so surprisingly, that people in Europe, I should say that this European Neanderthal sort of closer related than the Siberian Neanderthal to the Neanderthals that contributed to Europeans. And that then means that this genome allows us to identify more DNA in present-day people that come from Neanderthals because it's closer to the Neanderthals that actually contributed. So about 10-15% more Neanderthal DNA can be identified in any one person when you use this genome. Just to identify that, illustrate that, here is a version that's associated in present-day people with high levels of LDL cholesterol. So you may not want to have that version. This is this variant here. And I just indicate in black dots here variants that are identical to the Neanderthal, and identify the fact that it then sits in a chunk of DNA that is almost identical to the Neanderthal genome, over 130,000 bases. And it's this large size of these pieces that really shows that this comes over from Neanderthals quite recently, because recombination that happens when germ cells are formed in each generation makes these fragments smaller and smaller with time. So you could, of course, also have something where you're similar to Neanderthal just because you inherited from the common ancestor 500,000 years ago or so. But this big size of the chunks shows that it comes over back here, in this case and other cases we indicate. You could then go on and say people that live today closer to the Altai Mountains, to where this Neanderthal is found. Are they more closely related to this Neanderthal or that Neanderthal? And they turn out to still be closer related to the European Neanderthal, although they live closer to where this one is found. So to supporting this idea that a large part, anyway, of the Neanderthal contribution happened early on in Western Eurasia somewhere. There are also now several Neanderthal genomes of late Neanderthals, some more intermediate quality. There are 5 from around Europe that recently published. Much of the progress in this comes from then improving techniques, because as opposed to when you study the Neolithic expansion or something like that, where you can look at hundreds of skeletons and identify the best preserved ones, with Neanderthals, you're still quite limited in the number of individuals you can get study. So there are sort of improvements here that made this possible, that you, for example, increase the amount of endogenous DNA among the DNA fragments of sequence from 4% to 30% by certain treatments of your specimens or your samples. What comes out of this is that the late Neanderthals are all very closely related to each other. So there will be in the future some insight in Neanderthal genetic history, and it will involve, I think, the spread of Neanderthals rather late on in their history, replacing other Neanderthal groups. There is now a third high-quality Neanderthal genome that's not published yet. It comes from this site, Chagirskaya Cave, also in the Altai Mountains. Another site, the Denisova Cave, it's this place here. That genome is available on the web, so we made it, you can download it if you like, but we haven't published it yet. But if you go to this place here, this genome turns out, although it's found close to the Denisova Cave, it's more closely related to the Vindhya genome in Western Eurasia. It's in the order of 80,000 years old or so. So in the Altai Mountains then, 120,000 years ago approximately, there were Neanderthals there. later there were Neanderthals there that were more closely related to Neanderthals in Western Eurasia. So there will be some interesting insights in sort of Neanderthal population history that will be emerging from this. Yes, so as you also know then, in the Denisova cave, one also found this fragment of the pinky of a girl, and one could sequence a good genome from it. And we were quite surprised to find that it was not close to the Neanderthal, not a modern human, but something else that go far back to common ancestors shared with Neanderthals. So all these estimates in years are very unreliable. We're even still unknown, don't really know the mutation rate in humans accurately, actually. But the relative sort of estimates you can still do. So if this is something like 100,000, say, the deepest divergence between two present-day African populations, this is an order of 4 times deeper. So that's what we then call Denisovans nowadays. They are then only known from this one site, physical remains of them, from this little finger bone, 3 teeth that have been found in the cave, and now a small cranial fragments also that has recently been identified from the cave. But we think they have been much more widespread, or that have been quite widespread in Eurasia in the past, because we find contributions from the Denisovans to present-day people. We don't find it in Europe and Western Eurasia, but you find it all over mainland Asia, well below 1%, and you find a substantial contribution in people in Oceania. Up to 5–6% of the genomes of people in Papua New Guinea, for example, come from Denisovans. And not only that, Josh Akey has used our genome sequences and analyzed genomes from people in Asia and found that there must have been different Denisovan populations in the past. So these are analyses where they take genomes from people who live in Papua New Guinea today and plot then sort of genomes as dots on this and make this landscape where each individual is scored for how close they are to the Neanderthal genome here. So you see there is sort of parts of the genomes in Papua New Guineans that are very close to the Neanderthal and not at all close to the Denisovan genome, which is plotted here. And then there is this component here that's not close to the Neanderthal, but close to the Denisovan. So these are these 2 components that were contributed to Denisovans, to Papua New Guineans from Neanderthals first, and then from Denisovans here. But if you then look in genomes from Japan, you will find the Neanderthal contribution, as you would expect. You will find a Denisovan contribution that's tiny, as we said, on other parts of Asia, similar in its similarity to the Denisovan genome as this component here. But you find an additional component here that's actually very, very close to our Denisovan genome. So there are 2 independent Denisovan populations that have contributed to the genomes of people in Japan and actually China also. So this then shows that Denisovans, there were at least 2 divergent populations of Denisovans. And you find evidence of these 2 populations. You don't find it in India and southern Asia, but you do find it in China and in Japan, as I said. So there is sort of an interesting population history and substructure in Denisovans that we actually don't see in Neanderthals to that extent at all. So just to make a sketch for you then about what we think about the origins of Neanderthals and Denisovans from studying genomes. They have an origin in Africa more than 500,000 years ago at some point. Their ancestors then come out of Africa and evolve in Western Eurasia to what we call Neanderthals. And in Eastern Eurasia, we don't know where, just colored in all of it there. We call them Denisovans. We don't know where they have lived really at different times, but we know at some point in their histories there have been Neanderthals, and at some point Denisovans in this Altai Mountains in southern Siberia. Modern humans evolved from 300,000 years ago or so in Africa, spread to the Middle East, and start spreading seriously out into Eurasia somewhere between 50 and 100,000 years ago or so. Early on, they mix with Neanderthals, perhaps in the Levant or so. They continue to spread, and I think there is sort of good evidence that there is additional admixture in the ancestors of Asians. Asians today carry 20–30% more Neanderthal DNA than people in Europe. So instead of 1%, 1.3%, say. There is different opinions still, if that might be due to that the Neanderthal contribution was diluted in Europe from some population that had little or no Neanderthal contribution, or if there's additional admixture in ancestors of Asians. I tend to favor the latter suggestion. But then there is admixture in Asia, probably early on with Denisovans. There is at least one more admixture then from this other Denisovan population that contributes to ancestors of Chinese and Japanese. There is probably also additional admixture to account for the large component of Denisovan DNA in people in Papua New Guinea. It spreads out, these groups disappear and live on a little bit then in people today. In Africa, I should say, of course, modern humans appear somewhere in Africa and spread across Africa also. If one mixed with archaic forms outside Africa, one probably would have done it in Africa too. There is some indication of that from present-day genomes in Africans. We have no archaic genomes as yet from Africa, but I wouldn't be surprised if we will find something like that there too. So clearly the molecular data sort of rejects the total replacement. There is a contribution from these earlier forms of hominins to present-day people, well below 10% or so, but it does exist. And we're beginning to learn more about this in a very direct way, actually. The first indication of this came from Romania, from this site, Oase, and this mandible then. That is 40,000 years old. Looks, I understand, as a modern human mandible, but is one of the earliest modern humans in Europe then, at the time when there were Neanderthals around. So we were very interested in looking at if there was a Neanderthal contribution to this very early modern human. So this is just one chromosome. These are other modern humans, present day and ancient, where Neanderthal fragments are indicated along the chromosomes in color. And then the first one here is this Romanian guy that's 40,000 years old. And as you will see, he has dramatically more Neanderthal contribution than any other person we looked at so far, actually. And in fact, these things that are black here, we have no information. So more than half of this chromosome, we infer, is solidly of Neanderthal origin. So if you look across all the chromosomes in the genome here, we can find 7 such large chunks of large sections of arms of chromosomes that is from Neanderthals. So this, of course, indicates that this person had a close relative in his family history that was of Neanderthal origin. So if this is this person, of course, then has 50% of its DNA from its mother and father, 25 from a grandparent, and so on, back through the generations. And we can then show that this extent of contribution that we see means that 6 generations, 5 generations, or 4 generations back, there was a Neanderthal ancestor to this individual. And we can't say how many generations back it was, because it becomes very stochastic, how much you have from your grandparents. There is even a certain chance that you have nothing from any individual person 6 generations back in your family tree. So we sort of stumbled over someone here that has a close Neanderthal ancestor. And of the different contributions between Neanderthals, Denisovans, and modern humans that we've inferred then from studying the whole genomes, we then sort of stumble across one case of this, the contribution from Neanderthals to modern humans in this one case here. The other case of sort of more directly looking at mixture between groups is then from Denisova cave. And that sort of comes from work that we've done with Tom Hyam and Michael Buckley on screening many undiagnostic bones to find ones that may be of hominin origin. So as you know better than me, most bones you find, this is an example from Denisova cave, are these undiagnostic bones, fragments that you don't know if they are hominin, human or animal. Vast majority of them are animals. And we then use this proteomic technique that Carl has talked about, looking for collagen fragments in these things that allow you to then say, is this a hominin or is it what animal is it at a coarse taxonomic level? So it will not distinguish between modern humans, Neanderthals, and Denisovans, but you will be able to tell if it's a hominin. So then from screening 2,200 such fragments, they found one fragment that was of hominin origin. And we sequenced the mitochondrial DNA, and it was of Neanderthal. So this is this bone, it's about this big. But it was well enough preserved, so we went on and studied the nuclear genome too. And we're very surprised to find that it seemed the mitochondrial genome was totally Neanderthal. But the nuclear genome seemed to be equally related to Neanderthals and Denisovans. So if we plot in blue Neanderthal variants and in red Denisovan variants across each chromosome in the genome, you find that each chromosome is sort of painted blue and red in approximately equal proportions here. So, and if we then look at the amount of genetic variation in this individual, what I haven't pointed out is that if we now look on Africans and non-Africans today, Africans have more variation because the sort of overall origin of the gene pool is in Africa, one accumulated genetic variation there. Part of that variation colonized the rest of the world. And if you look at Neanderthals and Denisovans, they had even less variation. They seem to have lived in small groups and have 3 to 4 times less variation than non-Africans have today. But this individual is, of course, then a mixture between Neanderthals and Denisovans, so they have more variation, this one individual, about like Africans have today. And you can also say that the proportion of heterozygote sites is what you would expect from a first-generation individual, actually. So it turns out that you can then show that this individual has a mother that is a Neanderthal, because the mitochondrial DNA is Neanderthal, and a father that's Denisovan. This is sort of a first-generation individual from these two groups. And not only that, if you look carefully at this, there are 5 tiny little parts of the genome indicated in green here, and one of them is blown up here. There's a segment of the chromosome that looks like it's all Neanderthal. So if you compare to a good Neanderthal, or sort of good Neanderthal, a normal Neanderthal, this segment of the genome looks pretty much like any Neanderthal would. So this indicates that the Denisovan father then have some Neanderthal ancestry back in his history. So somewhere back here, the Denisovan father have some Neanderthal ancestors, but quite far back, in the order of 10, 20,000 years back. So of these different mixtures we have inferred from the genome, we then stumble across one of them here, a direct offspring of a Neanderthal and a Denisovan. So this sort of shows that these groups have always mixed with each other when they met, of course, gets very interesting to look more carefully at this type. Well, one thing I should also say then is that you can now, of course, infer some of the population history here from looking at the genome from the father of this individual, the Denisovan father. He is quite closely related to the Denisovan genome from the same cave. The mother is more interesting, actually. She is not at all closely related to the Altai-Neanderthal genome that from the individual that lived, that you found just a few meters away in the cave, but is a lot older. So we estimate that this individual lived somewhere in the order of 80, 90,000 years ago. The 120,000-year-old Neanderthal in the cave is not closely related to the mother here. She's more closely related to Neanderthals in Europe that lived 20,000 years later. Again, showing that there must have been interesting population movements here, either as a movement and from west to east or east to west. And we will eventually find out that, I think, when we have older Neanderthal genomes in Europe also. But you, of course, get interested in reconstructing the detailed population history at a site like this. And I have a good hopes one will be able to do that at this and other sites by studying sediments and DNA from sediments. So Viviane Sloan and Matthias have worked out the methods to quite efficiently retrieve DNA from just archaeological sediments where you don't see any evidence of bone fragments or anything like that in them. And I showed last year, for example, that at different sites you can then reconstruct the fauna that has existed in different layers quite well. And sometimes you can also retrieve DNA from the hominins that have lived in the cave and show if, say, Neanderthals or Denisovans have lived there. So you can put the evidence together, say, in this Denisova cave, and you will tend to deep down in the stratigraphy, find Denisovans and Neanderthals, Denisovans and Neanderthals again. And what you would like to do is do this in a very detailed way. But that then requires studying many, many sediment samples, of course. So what is then very useful is the sort of automation of this, where in our laboratory we have now automated the DNA extraction from sediments and bones. So robots like this do it, then produces DNA libraries and fish out the DNA fragments that you are interested in studying. So you can process hundreds of samples per week. And one graduate student, Elena Zavala, has taken this on in Denisova Cave then, just in the process of working on that, but she can then, what you can easily find is animal DNA in this. The hominin DNA is much rarer, but you can then find, say, Neanderthals here, and several samples, and Denisovans here, in this particular part of the stratigraphy. So she's sort of working on this at a large scale. A sort of question you have to address is, of course, leaching. How much can DNA move between layers in the cave? So it will be very cool. So what I really want to make propaganda for here is that you should collect sediment samples through your stratigraphies when you make excavations. You can store them for tens of years, and one can then in the future analyze them even if you don't do it now. And one will get better and better on this. But you should then sort of collect at least 5 or 10 samples per layer, let's say, because animal DNA is generally easy to retrieve. Hominin DNA is more, you have to look at many samples. And also layers, say, below that are sterile, archaeologically sterile, because one will be interested in sort of demonstrating that you then in archaeologically sterile layers don't find hominin DNA, because the extent of sort of how this can move through the stratigraphy is often an open question and may also be different at different sites. But you don't need to collect big samples. 100 milligrams of sediments is enough, but many samples. And of course, clearly logged where they come from in the stratigraphy. So, but to take a bigger view of this, then I am very surprised, I must say, that from the Denisova cave, we have only 6 individuals for which we have genetic information, and one of these turned out to be a first-generation offspring of a Neanderthal and Denisovan. If we look at modern humans that are old enough so they could have overlapped in time with Neanderthals, we have only 3 individuals who have genetic information. This 45,000-year-old individual from Western Siberia, 1 individual from outside Beijing, and this person from Huesos. And one of these 3 then turns out to have a close relative that is Neanderthal. You know, when we first found this, I said, well, sometimes you're lucky, particularly we are often lucky. But now we stumble across this, and you can't be lucky twice, I think. But it's, of course, also very hard to make statistics on so through numbers. But you sort of begin to wonder, since you find these individuals with close relatives from 2 groups so easily, if part of the story why Neanderthals and Denisovans disappeared may be that they were simply absorbed into modern human populations when they came. Now, that said, there can, of course, be different scenarios at different times and in different regions for what happened. It doesn't need to be just one explanation to this. But since we stumble across these sort of individuals, one scenario maybe, or a null hypothesis, is that one simply mixed with each other freely when you met. But there might have been more modern humans. We, after all, have only 2% Neanderthal DNA in present-day non-Africans, right? So, and that is something that has been suggested from the fossil record too. For example, Eric Trinkhaus, even before there were any any DNA at all, suggested that part of the story is sort of modest level of assimilation of Neanderthals into modern humans. Another interesting question then, this is then talking about the end of Neanderthals, if you like, and Denisovans, is their origin. And there is then only little thing we know about that. Carlos referred to already. It's a site, Sima de los Huesos, a steep cave that you probably are familiar with, where we found large numbers of hominins, and where we are struggling with retrieving DNA from it. It's around 430,000 years old, so it's 5 times older than most other Neanderthals remains we study. Just to give you a feeling for how much work this is. So for the mitochondrial genome, for retrieving a mitochondrial genome, we sequenced about 500 million DNA molecules, and around 15,000 of them come from the mitochondrial genome. So you sort of have to throw away 500 million molecules to have the ones you need. You can then retrieve a Neanderthal mitochondrial genome. It's here. And quite surprisingly, it is not close to the Denisovan mitochondrial genome, but shares an ancestor with it quite far back. It does not share a direct ancestor with the Neanderthal mitochondrial genomes. Rather, that shares an ancestor with present-day human mitochondrial genomes. So that is, of course, a bit disturbing, since we know that Denisovans and Neanderthals are in fact sister groups when you study the nuclear genomes. So 2 years ago, we then managed to retrieve a bit of nuclear DNA also from Sima de los Huesos. And most sites it shares with Neanderthals, much, much less with Denisovans and with modern humans. So quite clearly, in this scheme of things, Sima falls early, but clearly on the lineage leading to Neanderthals when you look at the nuclear genome. But the mitochondrial genome then shows a different picture. So where it's closer to the Denisovans. And one possibility to explain that would be if actually the mitochondrial genome in late Neanderthals comes from ancestors of modern humans at some point. So If this is a tree of the mitochondrial genomes of Neanderthals, here is Sima de los Huesos that shares an ancestor with Denisovans. This would then suggest that the mitochondrial genome comes over from an ancestor of modern humans into an ancestor of late Neanderthals, somewhere between these dates here, or these branches in the tree. So one suggestion is that this sort of indicates gene flow from Africa into Neanderthals somewhere between 200,000 and 400,000 years ago or so. And we are now working quite hard to retrieving more nuclear DNA from Sima de los Huesos to maybe address that. Maybe we should then be able to see evidence of this gene flow from Africa in later Neanderthals when we compare it to this Neanderthal ancestor 400,000 years ago. So I think we will learn a lot more about the population history of Neanderthals in the next few years. But before ending then, I wanted to talk a little bit about the functional implications of this, of the contribution of Neanderthals to present-day people. I think that's almost one of the most interesting aspects is what we can learn from the genomes about physiology of Neanderthals, how it affected people today, and what may be unique to modern humans. So you can, of course, walk over the genomes of, say, Europeans today. This is the whole genome, plotting the frequency with which you have things from Neanderthals in windows. And you will see that's 1 or 2% or so, and then sometimes there are things that many people have. 60% of Europeans in some region have something from Neanderthals. So that suggests that something has come over and been positively selected. So it rose in frequency, so many people got it. Just show you 2 examples. We know of several of these things now. This is one of the earliest things that was found. It's a part of the genome that up to 25% of people in East Asia and in Native Americans have, not in Europe. It clearly comes from Neanderthals in those individuals, and it encodes a lipid transporter, so a transporter molecule that pulls fat into your cells in your body that has several differences in the Neanderthal version to the modern version that most people carry. And carrying this variant then infers bigger risk of getting type 2 diabetes, a type of diabetes you get at old age. So sort of maybe surprising why something that gives us a disease or contributes to disease today have risen to high frequency. But I think there's good reason in this case to speculate that things that give us diabetes today when we eat almost too much all of our lives may actually be advantageous in another situation when you have periodic starvation, because it sort of makes you more conserve more of your energy, and that's sort of good in starvation and bad when you have too much to eat. This may be some adaptation to starvation. Just last month, there was a version of the progesterone receptor, a receptor for a hormone that's important in pregnancy, that is at quite high frequency in Western Eurasia, that comes from Neanderthals, and turns out to be associated with preterm births, with having premature babies today. It's of course very hard there to think out some evolutionary story why that would have been advantageous in the past. But I think it also goes to say that most genetic variants have several effects, and we just know some of them, generally from medical studies where you find an association with some medical problem, but they may have other beneficial effects that you sort of don't know about. Have Denisovans contributed to present-day people? Yes, they have. The most famous example is this adaptation to high altitude in Tibet, where there is a version of a gene called EPAS1 that's 80% frequency in Tibet. It allows you to absorb more oxygen in your blood when you live at high altitudes where there is little oxygen in the atmosphere. to do that without doing this adaptation that other people do when they move at high altitudes, where you have many red blood cells, because that leads to complications with blood clots and things like that. So in Tibet then, Rasmus Nielsen's group showed that in Tibetans, this version of this gene comes over from Denisovans into the ancestors of Tibetans. There are other things from Tibetans too. There is a version of a gene cluster where some genes have to do with brown fat that's particularly important in generating heat. This version is almost fixed in Inuits. Sort of all Inuits have it. It exists at several other places in Eurasia. And this version, so this gene over here, is very close to Denisovans, much further from Denisovans, and much, much further away from everyone else. So this gene cluster has clearly come over from Denisovans, and it may have something to do with heat generation, sort of making me think about the interesting talk we heard yesterday. So some things seem to have been so important that they come over even twice from both Neanderthals and Denisovans independently. One example is a cluster of genes here, toll-like receptors. They sit on the surface of immune cells and are important for innate immunity. Immunity that you have from birth against viruses and bacteria. This has come over both from Neanderthals and Denisovans, risen to high frequency. And you can show that people that carry two versions of the Neanderthal express more of these receptors on the surface of their cells than those that are heterozygous, have one modern and one Neanderthal variant, or two modern variants. And in association studies, you can show that having these Neanderthal and Denisovan variants sort of protects you against ulcers, because it protects you against infection with Helicobacter pylori, the bacterium that gives you ulcers. The very same variants, though, gives you higher risk of having allergic problems. So if you have allergies, you should perhaps blame Neanderthals, but if you don't have an ulcer, you can sort of thank them for it. So there is now quite a lot of things indicated in red here, variants that seem to have been positively selected that came over from Neanderthals in this case. Many of them have to do with immunity against infections, others with skin and pigmentation. There are other examples too, as you heard. Most things we know of, though, are associated with diseases. I think that's sort of really ascertainment. This knowledge comes from association studies where people look for genetic variants associated with disease. But there are things involved with blood clots, with urinary tract infections, many skin diseases, depression, and so on, that come from archaics. In general, though, I think this is not just ascertainment. I think overall there seems to be negative selection against variants from Neanderthals. And for example, Josh Akey's group at Princeton has shown that if you look at people that carry then two variants of a gene, one from mom and one from dad, one comes from Neanderthals and the other one is a modern one, and look at the expression of these two genes, for example, in the brain, you would expect, the null hypothesis is you expect as much of your mom's version as your father's version, 50-50. But here is an example of a gene expressed in the brain that where you don't express it 50-50, you express less of the Neanderthal version. And overall, they find, when they look in many different tissues here would be 50-50. If you're below this line, you express less of the Neanderthal version. Overall, you really express less of these Neanderthal genes. And that's particularly prevalent in the brain and in the testis for some reason. There is sort of evidence, I think, of negative selection. We recently just looked also at, if you look over the whole genome, you have a little over 2% Neanderthal variants. If you look in different parts of the gene, the one part that stands out is this regulatory part where you have significantly less Neanderthal contribution, about half of what you have overall in the genome. Again, suggesting that there's something with regulation of genes that is sort of bad in the modern human background overall. Finally then, I want to sort of raise a more controversial issue of sort of saying one interesting thing may be to study what did not come over from Neanderthals. What are we sort of resistant against? So you can go over the whole genome, chromosome by chromosome, plot again in red, frequency of Neanderthal contributions in red, in Europe in red, and in Asia in green. And you can ask, are there parts of our genome where we statistically would expect Neanderthal contributions, but we find none? And there are such regions. Particularly cool is to look in Papua New Guinea, say, because there you have a lot of archaic contribution. You have first this contribution from Neanderthals to all non-Africans, and you have this additional contribution then of 3-4 times more from Denisovans. And you can look for regions such as this on the chromosome then, where you find neither Neanderthal nor Denisovan contribution in a sort of part of the genome, and say, what could be hiding in there? So things then, we think that that may be part then where some function is hiding in the genome that's unique to modern humans, that make us select against the contribution from these other groups. So things that changed here and might be important. And of course, we sort of think that modern humans, at least I think, are quite special when you compare to these other groups. After all, only modern humans with time develop technology and culture that change really rapidly and develop art. There is now evidence of Neanderthal art, but figurative art that really shows immediately you see what it depicts comes only with modern humans. And all the modern humans become millions of people and colonize the whole habitable part of the planet. There is something, I believe, special with the sort of ability to develop culture and technology in modern humans. So a sort of dream would be that that has the readiness to develop that and accumulate human culture across sort of generations, maybe due to changes here. So we can, of course, list all genetic changes now that happen here, exist in everyone today, no matter where we live on the planet, but we're not there in Neanderthals. And that's a small list of things. It's something like 30,000 genetic changes. So you can actually look through them in a computer in an afternoon, but you have no clue what you look at. That's the problem. We are very, very bad at inferring the function of any particular change in the genome that we're interested in. I just want to end by giving you a flavor of how we're trying to approach this. And in the most simple part, perhaps, of some type of changes, those that change amino acids in proteins that actually then perform functions in our body. So those proteins that have such changes are just 87 such genes. You may ask which ones are important. Our bias is, of course, to think it should have to do with cognition, with the sort of development or function of our brain. See, I'm running out of time, but we've then sort of focused on things that changed on the human lineage, and particularly using data from Philip Kaitovic's lab, a previous postdoc who's now in Moscow, who studied the metabolism of the human brain and look for changes in metabolite in small molecules where humans differ both from chimps and from macaques, the things that are unique to humans. And one pathway that stands out, only two pathways that stand out, is this biochemical pathways where you make G's and A's, two of the bases that occur, for example, in the DNA. So these are called purines, and there are less of them in the human brain than in the brain of macaques and chimps. So they know that it changed somewhere along this lineage from the common ancestor of the chimps and other apes. But looking at this list, we then are struck by one enzyme here, ADSL, because this enzyme catalyzes 2 steps in the synthesis of these 2 bases, A's and G's. And not only that, that amino acid change is close to the active site of the enzyme where it really does its job. So we have put these changes into the mouse, so made the mouse more similar to the human. We then find a reduction in different tissues actually in the mouse of this enzymatic activity, and where we have the same metabolites we can detect, they go in the same direction, down. That's still in the mouse, but it's sort of a direction then where we'll go in the future. I think Neanderthalizing mice and humanizing mice to study things such as metabolism. But you might want to do this also in the human genetic background, of course. So what you can then do is engineer in this one change in human cells back to the ancestral Neanderthal-like states. So we've done this in this case, changing this one amino acid to the other, and now have cells that for this enzyme are like the common ancestor of Neanderthals and modern humans, and like the Neanderthals. We can then look at the metabolism of these cells, of course. And what we find is that all these compounds that are downstream of this enzyme in the biochemical pathways are reduced in concentration in these ancestralized cells. So I think we will actually be able to reconstruct things, particularly that has to do with metabolism in the future. We can now, on top of this, add in more of these changes, ancestralize these cells for several parts of its metabolism, and actually study it in living cells. So I should end just to say that I think one of the most interesting things for the future then is to study the physiology that changed here on the human lineage. That changed here. In the future, when we have many Neanderthal genomes, we're already beginning to be there. We will also be able to things that changed on the Neanderthal lineage to actually study parts of the physiology that's unique to Neanderthals. And the way to do that will be to engineer these things into human stem cells, say. Stem cells that you can then differentiate into neurons, into liver cells, into different types of cells, and study their metabolism. And I think you can also sort of humanize and Neanderthalize mice and other model organisms. Many people helped with this, many more than I can mention. Many paleontologists contributed samples. Many people helped analyze the genomes. Particularly, I mentioned Monty Slatkin at Berkeley and David Reich at Broad that helped with analysis. Many people are involved in functional work. I show the current group leaders in our department, particularly Matthias Meyer, who I already mentioned, who developed technology for retrieving ancient genomes, and Janet Kelsey, who does all the bioinformatics, and people we collaborate with, particularly Philip Kajtovic, then on the metabolomics, and Philip, this person. And with that, I thank you for your attention.